Hello everyone, my name is Jordon Inkol. I'm a second year master's student at the Ontario Veterinary College, and I'm going to be presenting my project on Cannabidiol radiosensitizing canine urothelial carcinoma cells as a naturally occurring model of muscle invasive bladder cancer in vitro. So just before I begin, I just want to give a quick funding disclosure. So I want to thank the OVC Pet Trust as well as Grey Wolf Animal Health for providing funding for this project. To begin, I'll be doing a quick introduction on canine and human urothelial carcinoma. So, urothelial carcinoma, or UC, as I will refer to it now on my slides, comprises about 1 to 2% of all cancer diagnoses in dogs, is the most common form of bladder cancer in dogs. Clinically, it presents with bloody urine, increased urination frequency, as well as discomfort or straining during urination. In dogs, it typically presents as high-grade muscle invasive, with about 57% of dogs having distant mets at the time of necropsy. As you can see in the figure on the left-hand side there, those kind of orangey blotches are where typically the tumors form in dogs. Now for humans, it's typically divided into low-grade non-invasive, which is the more common form, and high-grade invasive, which is the least common form. Both canine urothelial carcinoma and human high-grade invasive urothelial carcinoma share histological and molecular markers which make them very similar diseases. So typical treatment for canine UC is systemic medical therapy, which includes some form of a chemotherapy agent in conjunction with a nonsteroidal and anti-inflammatory or NSAID. Surgery and radiation are typically not feasible. Surgery runs the risk of spreading the tumor further within the actual abdomen of the dog, as well as typically the location of the tumor itself within dogs is inoperable. And then for radiation, typically dogs just don't respond too well currently, but we're hoping to improve that with this project. And typically the median survival is 8 months for these dogs. Now, in the case of humans, typically surgery and adjuvant chemotherapy or immunotherapy are considered mainline treatments. And radiation therapy is typically only reserved for palliative care, just to decrease the pain and kind of tumor burden. But unfortunately, 50% of these patients will still succumb to their disease if they have the high-grade, high-invasive form. So now since this project revolves around radiation treatment, I thought I'd talk about just kind of some basic things about it. So radiation doses are defined as gray or GY. Typically they're administered in fractions or small doses essentially. So 2 by 10 gray would be 2 doses of 10 gray, or in this case 10 by 2, which is 10 doses of 2 gray. It's typically used to induce DNA damage in cancer cells, which in turn would cause cancer cell death. And then the 4 things to keep in mind, or the 4 R's rather, when looking at response to radiation therapy is repair of DNA damage, redistribution of cells in the cell cycle, repopulation, and then reoxygenation of hypoxic tumors. So essentially what this means is repair of DNA damage is how do the cancer cells actually repair the damage induced by the radiation treatment. Redistribution of cells in the cell cycle is essentially cells that are in more favorable parts of the cell cycle will die first, whereas those that are in the least favorable will die last and later on in the fractions. Repopulation is this phenomenon of surviving tumor cells being able to proliferate during fractionated radiotherapy. And then reoxygenation of hypoxic tumors is when hypoxic tumors become reoxygenated during, or between rather, fractionated radiotherapy. So now I'm going to just talk about cannabidiol and rather its use within cancer treatment itself. So as most of you will know, CBD is one of the most well-studied phytocannabinoids in cancer research alongside THC. Currently for human oncology services, it's used to treat things like nausea, as well as stimulate hunger for patients undergoing active chemotherapy. It's currently not approved in veterinary oncology for any use at all, but some owners will administer it to their pets on their own. Currently, there's an in vivo human orthotopic glioma model that shows that CBD may act as a radiosensitizer. So we sought out to see if this radiosensitization effect of CBD shown in this study would work for canine and human urothelial carcinoma, but using canine urothelial carcinoma cell lines as a model for the human disease. So our hypothesis was that CBD exposure will act as a radiosensitizer in canine urothelial carcinoma cell lines. The double-stranded breaks from radiation damage will be increased in those who are exposed to CBD, as well as CBD will potentiate the reactive oxygen species or ROS generation from radiation therapy. I'll now move into our materials and methods for this study. For this study, we used 3 canine urothelial carcinoma cell lines, AXA, ORIG, and SH. We then mainly used a clonogenic survival assay, and how it works is we seed cells into a 6-well plate, we expose them to vehicle or CBD, and then they undergo radiation treatment from 0 to 10 gray. Then we used the survival data from that assay and plugged it into our regression formulae, where we used a linear quadratic as well as a nonlinear log logistic, and I'll get into why we did both later. And then using dimensional reducing techniques, such as principal component and hierarchical analysis, we're able to take these survival and extract radiosensitization scores from them. Then we also use gamma H2AX immunofluorescence, which is a marker of double-stranded breaks. And then we just used a reactive oxygen species assay kit. So getting into the results, when we plugged our survival data from our clonogenic survival assay into our linear quadratic formula, we were able to get the graphs shown in A, and as you can see, there's a marked difference between the cells that are exposed to vehicle or CBD. And then extracting the actual parameters from these models and putting them into the table shown in B here, we can see that the alpha parameter actually increases. And there's studies showing that as alpha increases, our biologically effective dose decreases. And our biologically effective dose, or BED, accounts for things like total dose rate, total treatment time, as well as dose per fraction. And what this means essentially is that as alpha increases, our BED decreases, meaning our cells are more sensitive to the radiotherapy. And then moving on to the beta parameter, it also increased, which means they would affect the actual alpha beta ratio. And as the alpha beta ratio decreases in this case, it means that they're more sensitive to a higher dose fraction regimen. But because the linear quadratic parameters aren't very easy to interpret from a biological standpoint, we employed the use of a nonlinear log logistic, as these parameters are a lot easier to interpret from a biological standpoint. So our representative graphs are shown in A here, and then extracting the parameters from them in B, we can see psi1, which accounts for the survival fraction as the dose approaches affinity. Psi2 is the ED50, or rather the radiation dose, in which the survival fraction is 50%. And then Psi3, which just accounts for the steepness of the curve. And as we can see here in Psi2, there's a drastic change in the ED50 when cells are exposed to CBD, and in most cases it's a 0.5-fold change, so it's halving the dose, which is really indicative of a sensitization. And interestingly enough, the slopes actually decreasing, they're not as steep, potentially meaning that this decrease is drawn out over a longer period of radiation doses. And then as we can see in C here, that there's actually potentially a clustering of cells as they're exposed to CBD towards a more radiosensitive, but it's a bit hard to interpret. And that leads me into our next method, which was using something called principal component and hierarchical analysis to really truly see if these cells were becoming radiosensitized and that we could group them into distinct radiosensitive and radioresistant groups. We employed these methods. So in A, we have principal component 1 and principal component 2 plotting against each other, and we essentially chose to do these because they explain over 98% of the variance within the data set, and we set a cutoff of 90%. So as we can see there, the cells that were treated with CBD clustered together really nicely into these radiosensitive groups compared to the vehicle treated ones, which were spread out over this kind of large radioresistant group. And then using principal component 1 and plotting it against our AD50 or Psi2 here, we can see there's a really great correlation between the two variables, getting a Pearson R of 0.9263, suggesting that our principal component 1 is really good at predicting whether cells are radioresistant or radiosensitive, regardless of the dose from 0 to 10 gray. So then using this data, we used the hierarchical analysis to see if we could really distinctly group these treatments into radioresistant and radiosensitive, which is shown in D. As you can see, we get a really good clustering of our CBD treated cells and our clustering of our vehicle-treated cells, suggesting that the CBD does actually radiosensitize all three cell lines. And to confirm that these clusterings were proper, we use a silhouette plot, which is shown in E. And essentially any width that is greater than 0.5 suggests a strong fit. And as we can see, all of our fittings look accurate. So then moving on to see how the CBD was actually influencing the cell on a molecular basis, we looked at gamma H2AX, which, as I said before, is a marker of double-stranded breaks. So we just have some representative images on the left here, showing that when we treat with CBD, we get this increase in fluorescence in our cells, suggesting that there's an increase in double-stranded breaks. This image is taken at 6 gray, so that's why the vehicle actually shows some double-stranded breaks as well, because radiation on its own will induce it. So then we took how intense the fluorescence signal was from each cells and averaged them across 4 images. And that's what we graphed in B here. It's called the CTCF, which is the corrected total cellular fluorescence. Essentially what we were able to conclude is that CBD within the same dose of radiation, so comparing 8 gray vehicle treated and 8 gray CBD treated, we saw an increase in the amount of double-stranded breaks. But there appears to be no dose-dependent effects, meaning that from 6 to 8 gray, we saw a significant increase, but from 4 to 6 gray, there wasn't a significant increase between CBD-treated cell lines. So now we want to evaluate to see if reactive oxygen species was increased in our CBD-treated cells, kind of potentiating the effects of the radiation therapy. So as we can see at 0 gray, CBD can induce ROS production on its own. And when you combine it with radiation, it kind of potentiates its effects, except for in 2 gray, in which we actually surprisingly weren't able to see a difference between our vehicle and CBD treated. The reasoning for this, I don't know for sure. Also, interestingly, SH showed only significance at 0 gray and 6 gray. Potentially, this could be SH is a metastatic cell line, so it may have some mutations that are more radio-resistant than the other two. Now we were curious about what specific species of reactive oxygen species we were seeing an increase in here. And there's a good paper showing that radiation on its own can increase mitochondrial hydroxyl radical reactive oxygen species. So we set out to see if we were seeing an increase in this as well. And surely enough, in all the doses that we saw an increase in ROS on the previous slide, we saw an increase in this mitochondrial hydroxy radical generation, suggesting that this is the species we're seeing an increase in. So just kind of like the take-home message to wrap it up. So CBD radiosensitizes canine urothelial carcinoma cell lines in vitro, which can act as a good model for the human disease. It does this by enhancing double-stranded breaks as well as increasing reactive oxygen species generation, specifically those mitochondrial hydroxy radicals I talked about. So just some future work plans for this is to confirm that these effects actually result in apoptosis or programmed cell death. And then looking at protein signaling for DNA damage, especially in the SH cell line, to see if there's any proteins associated with DNA damage repair that are mutated. So with that, I'd like to say just a quick thank you to my advisors, Dr. Sam Hawker and Anthony Mutzers, my brother in the McPhee lab for helping with all the mathematical modeling, the radiation oncology team at the OVC, Laura Furness and Dr. Valerie Poirier, my fellow lab members in the Mutzers lab, and then I want to thank Dr. Deborah Knapp at Purdue University for providing the cell lines for the studies. Any questions about the study or further inquiries, just email me at jinkall@uogulf.ca. Thank you.